Right. Good afternoon, everyone. Good afternoon to online and physical students. So we are at the last part of cellular respiration now. So we discuss glycolysis only, right? Okay. Continue. So did we write the description of glycolysis? Virat? Okay. Right. Then write the next topic. Pyruvate oxidation and Krebs cycle. Pyruvate oxidation and Krebs cycle. Right, subtopic again, pyruvate oxidation. Right, listen now. This is what happening. So we have pyruvate. How many carbon are there in pyruvate? 3 carbon compound. So 3 carbon pyruvate is converted into, transported into mitochondrial matrix now. So the pyruvate oxidation and Krebs cycle, the location is what put there? Mitochondrial matrix, matrix of mitochondria. So using some amount of ATP to ATP, the pyruvate is being transported to the matrix of mitochondria. Now pyruvate is at matrix of mitochondria. It's being converted into acetyl coenzyme A. Two of these will produce two acetyl coenzyme A molecules. Acetyl coenzyme is a 2 carbon compound, so that it is releasing what put 2 carbon dioxide molecules, and also it will be reducing NAD plus into NADH. Hari, understood? This can be written as an equation also. They say that initially the pyruvate is converted to acetyl group by releasing 2 carbon dioxide molecules. Then the acetyl group combines with coenzyme A to produce acetyl coenzyme A. So it is written like this: 2C3H4O3. This is pyruvate. You can write pyruvate also. Plus 2 coenzyme A plus 2 NAD plus will generate 2 acetyl coenzyme A plus 2 CO2 plus 2 NADH. Another, right? So this is summarizing the whole thing here. Sometimes they are questioning this part also. Okay? Right, write down this one first. It's okay. Harapati, I was describing the pyruvate oxidation. 3-carbon pyruvate is converted into 2-carbon acetyl coenzyme A at the mitochondrial matrix. In order to do that, the pyruvate should be transported from the cytosol to the mitochondrial matrix, right? So this equation summarizes what happens here. Sometimes they question this equation also, right? Okay, write it quickly. Others, go to the description there. Yeah. In the presence of oxygen only, the pyruvate oxidation and Krebs cycle occurs, right? Others, go to the description and read it once and see if you can understand. Good afternoon. Next week, you will be having the second part of the examination, okay? So be ready for that 2 hours paper, okay? Second part. Yeah, 2 hours. Having 2 essays, 2 essays and no, as I remember, 4 structured essays. Have it. Done reading? This conversion, yeah, it is enzyme catalyzed. You're asking enzymes are, no, enzymes are not. Previous content only that we discussed from mitosis, meiosis, ATP, né? Those things only now we took. And one before meiosis, mitosis, cell organelles, that's all, right? Another test is there from photosynthesis, respiration. That is also coming up. Yeah. Once I'm done with this, I'll be having that in vacation time. No. I was telling that content, cell organelles, mitosis, meiosis, ATP. Okay, cancers, galls, also. Right, done. Right. Now read and see if you can understand that part quickly. Who came now, just explaining what is happening in the mitochondrial matrix after the glycolysis. The pyruvate is being transported to the mitochondrial matrix using some energy, and then pyruvate is converted into acetyl coenzyme A. This is a 3-carbon compound. This is a 2-carbon compound. So carbon dioxide is released here, and also NAD plus is reduced here. This complete equation also the same thing. Sometimes they question this last equation, right? Understood? Now copy it to the writing book quickly. Write the description into the book quickly. Don't take a lot of time. We have to finish this. Acetyl coenzyme. Yeah, there is some intermediate steps here. They are not writing that one. Look at the picture given to you with the Krebs cycle. They are writing only this one, but they say that after conversion into acetyl group, then only combining with coenzyme A. Yeah. No need to know the intermediate. Just remember that coenzyme A combines with acetyl group. Write the description quickly. Yeah. So no one is asking the equations other than in the structure they say. You have to write in words. Especially the carbon numbers you should mention. If there is an enzyme you have to mention, right? Adding one more thing here. Okay, all right. Try the next topic, Krebs cycle. Krebs, K capital, is a person name. Alright, now everyone, are you all done? Right, so here, now we have acetyl coenzyme A. How many carbon? The 2-carbon compound. Acetyl coenzyme A combines with the 4-carbon compound oxaloacetic acid, which is a 4-carbon compound. We know about oxaloacetate. Where did we meet oxaloacetate? In the photosynthesis, C4 photosynthesis, right? So here, these two are combined to form citric acid. How many carbon should be there? 6-carbon compound. So this 6-carbon compound is converted back into 4-carbon oxaloacetate using some enzyme catalyzed reactions, right? There are so many intermediates we are not learning. Just remember that 6-carbon compound is converted to 4-carbon. So when 6-carbon is converted to 4-carbon, how many carbon dioxide should be released? 2, right? Remember very well, even if we produce 2 acetyl coenzyme A from 1 glucose molecule there, from the 2-pyruvate, only one is fed to the cycle at a time. So you have to have 2 cycles per 1 glucose molecule in order to complete the reaction, right? So here, they have found out. I also didn't do any research about it. They have found out, they are telling. Right, so here, carbon dioxide, 2 carbon dioxide will be removed at different places, right? So here, in this reaction, 1 ATP is produced. Per 1 cycle, okay? Also, this is producing FADH2 also. FAD plus H plus electron, you will get FADH2. Correct? And here, 3 NAD plus 3 H pluses, sorry, 6 H pluses plus 6 electrons will give you NADH, 3 of these, right? So this is not exact places I am drawing. I am just drawing a summary diagram which you will need only the summary. No need to know what are the intermediates here and what are the enzymes catalyzing each of these reactions. Remember very well, each of these reactions are catalyzed by specific enzymes. They know the names of the enzymes. They know the intermediates, right? So if you are doing further studies in biological science stream, you can get to know what these are. Luckily, you don't have these things. So you are having less stuff to study. Okay. Right. So here, yeah, this is what happened in the cycle. So I'm making a summary of Krebs cycle here. Over there. Not working. Turn off. Right. So here, summary for 1 cycle. I'm asking how many ATPs are produced, how many NADH are produced, and how many FADH2 are produced, and how many carbon dioxide are produced. Then per 2 cycles. 2 cycles meaning per 1 glucose. Understood? Right. Now you guys tell me How many ATPs per one cycle? Others can see. Wait. Can see now, ne? Right. One, ne? Then per two cycles, two ATPs. How many NADH per one cycle? Three. Two cycles, six. FADH2, one. Per two cycles, two. Carbon dioxide, understood? Right. This cycle is also known as the Krebs cycle. Another name I'm going to write. Also known as citric acid cycle, because the first product is citric acid. Why it is known as Krebs cycle? The reactions of this cycle have been found out by a scientist called Krebs, Hans Krebs. And then also this is citric acid has this kind of groups, carboxyl groups. Three of them are there, so that we call it as tricarboxylic acid cycle. Also, its shortened form TCA cycle is also the same. Understood? Right. Don't write anything. I just want you to go to the note and just read it and see if you can understand. They have found out. I don't know the reactions. They have done researches. Hmm? Yeah, that's a separate reaction. This is a separate reaction. Hmm? No. No. Did you read? Right. Now draw. Draw the cycle first. And then write this one and the description, right? You need to write the given description in the book. Adipati, are you done? Right. So there's a request to explain the summary table one more time. I'll be explaining it one more time. So here, this is one cycle now. So the citric acid, 6-carbon citric acid converted into 4-carbon oxaloacetate. So during the cycle, from the energy released by oxidizing the substrate, they will be producing 1 ATP per one cycle. So here it is showing 1 ATP here. And then 2 ATPs are for 2 cycles. Understood? Per 1 glucose, 2 acetyl coenzyme A is produced. So I told that 1 is fed to the cycle at a time. So this is happening twice for 1 glucose molecule. Can you understand? Right. So here also from the H pluses and electrons released from the substrate, 3 NADH is produced per 1 cycle. So 3 is per 1 cycle, per 2 cycles 6. And then FAD, H2, only 1 molecule is produced per 1 cycle, per 2 cycle it's 2. And carbon dioxide, 2 is released per 1 cycle, 2 cycles is 4. Right? So we are going for another summary table. You guys need to fill that one. So I'm summarizing glycolysis, pyruvate oxidation and Krebs cycle all together. So the number of ATP, carbon dioxide and FADH2, NADH you have to fill there. After 2 cycles, okay, per 1 glucose molecule, after 2 Krebs cycles are happening. So you have to consider the amount after 2 cycles, okay. You can write with me. Draw and fill the table quickly. Can't Hmm? Oh. Wait, we'll write 2 net because 2 is used. No? Only 2 is here. 2 net. Net word should be there. Okay. Finish. 2 after 2 cycles. Okay. Can we discuss? Right. In the glycolysis, how many ATP? 2 net. So, in the glycolysis, how many NADH are produced? 2. Are we producing FADH2? No. Carbon dioxide? No. Pyruvate oxidation, ATP production? No. NADH? 2. FADH2? 0. Carbon dioxide? 2. Krebs cycle, how many ATP? 2. NADH? 6. FADH2? 2. Carbon dioxide? 4. Let's get the total here. So here 4 ATP, 10 NADH, and FADH2 2, and carbon dioxide 6. So if you can remember, in the big equation we are writing 6 carbon dioxide is released. So here 6. This is how it has come, right? There's another thing to tell. The ATP production in glycolysis and Krebs cycle we call it as substrate level phosphorylation. Why it is called as substrate level phosphorylation? Yeah. So it is taking energy by oxidizing complex molecules, no? Right. So remember that as well. Right. Let's go to the electron transport chain. Write the topic, everyone. Electron transport chain. Give me another name for electron transport chain. Oxidative phosphorylation. Where is it happening? Cristae of mitochondria. Right. So if you can remember now, the energies only they are in 4 ATP molecules and 10 NADH molecules and 2 FADH2. Energy is not converted into ATP much in the first two steps. Now, in the third step only, the energy is going to get released from the molecules such as NADH and FADH2, right? So here, series of oxidative and reduction reactions happen in order to release the energy from the substrate, which is happening in the cristae of mitochondria. There are a lot of enzymes in the cristae of mitochondria in order to oxidize these molecules to get energy. For example, NADH is converted into NAD plus H pluses and electrons, and FADH2 is converted into FAD plus H plus electrons. So these energy which is released by oxidation of these molecules, we are producing ATP. So that is why it is called as oxidative phosphorylation. So they say that from 1 NADH, 2.5 ATP is produced. Are we having like half of the ATP or something? No. What they have done is they have taken the total number of ATP and divided it, right? These numbers are rather different from previous syllabus, so you can't do the questions without the new syllabus thing. The previous questions having answers different from this one, okay? So 1 FADH2 will give 1.5 ATP. Therefore, how many NADH we have now? For 10 NADH, how many ATP is going to get produced? 25. 2.5 into 10 now. So 25 ATP is going to get produced. Then we have 2 FADH2. What is going to happen? 1.5 into 2, 3 ATP. So altogether in the electron transport chain, how many? 28 ATPs are produced. So this is the highest amount of ATP, right? Hi, write down this one quickly. Just to say that oxidative and reduction reactions are happening, no need to draw it also, no issue. Correct? No need to balance. Just leave it. Now listen everyone. Now we have H pluses and electrons removed from the NADH and FADH2. So they are going to give these electrons to atmospheric oxygen, the molecular oxygen. H pluses plus oxygen plus electrons will produce water. Two of these is here, four of these, four electrons, right? So here is the final electron acceptor of the cellular aerobic respiration. What is it? Molecular oxygen. Correct? Write that too. Hmm? At the end of this, these electrons and H pluses taken by molecular oxygen to give water. So we are writing in the big equation 6 water molecules. Here it is. So now after electron transport chain, we are writing the summary in the cellular aerobic respiration. Right. Now you guys need to tell me how many ATPs are produced altogether. In the glycolysis, we took it as 2. Ne? ඊළඟට Krebs cycle 2. නේද? Then electron transport chain, how many? 28. So altogether, what's the total? 32. Right. So they say that 32 is correct for cells other than liver cells and cardiac cells. The liver cells and cardiac cells are not using 2-ATP to transport the NADH produced in the cytosol to bring it to the mitochondrial matrix. But all other cells are using 2-ATP. Therefore, 32 minus 2 will give 30 ATP in total in other cells, right? So this is for liver cells, cardiac cells. And other cells, this is the total now, right? Write these two also.
Now come and read the lesson in that section. Read and copy the note into the writing book.
Thirteen, given. Read the book, really. Active cells such as liver cells, cardiac cells having 32. It's not using 2 ATP. That's how no it is given. Yeah. Read it, read it. Other cells use 2 ATP to transport NADH to the mitochondrial matrix. That's why they have 30 instead of 32. Okay? Read the sentence what they have given. Loud, loud, I can't hear anything. Hmm. Hmm. Yeah, other cells only use 2 ATPs. Okay?
that numbers. No, wait, I'll explain it. All done? No need to write the numbers thingy, right? About substrate phosphorylation and all they have given. Everyone, you're done? Understood? Finish. Right. Do not get confused with the page number 69 that they have given the few numbers there. No? So I'll explain what it is given here. So they say that in the glycolysis, 2 ATPs are produced due to substrate level phosphorylation. Also, the glycolysis is producing how many NADH? 2 NADH now. So 2 NADH means that 2.5 into 2. So 2.5 into 2 means you will get 5 ATPs from oxidative phosphorylation in the electron transport chain. These ATPs are produced in the electron transport chain, but from the NADH produced here. That's why they have given, this is kind of a bit confusing for children. Yeah. Each step from the NADH amount that they produce, they have calculated how many ATPs are produced in the oxidative phosphorylation. Right, let's go to the pyruvate oxidation. Pyruvate oxidation also produces 2 NADH only. So from 2 NADH, it is producing 5 ATP from oxidative phosphorylation. If we go to Krebs cycle or citric acid cycle, we have 2 ATPs produced from substrate level phosphorylation. We have 6 NADH. So 6 into 2.5, how many? 15, no? 15 ATP from oxidative phosphorylation. And 2 in FADH2. 2 into 1.5, 3 ATP. So altogether from oxidative phosphorylation, 18 ATPs are produced from the results of Krebs cycle. And then electron transport chain, you know that 28 ATPs are there. So they have elaborated this in this way. Understood that one? No need to write it anywhere. Right. Everyone write the topic, anaerobic respiration.
Online students, just wait for 2-3 days. I didn't give the complete set of tutes to the class. For the physical students, today you'll be getting the first tute only. The other tutes I'll give by next week. Okay. For the next unit 3, anaerobic respiration. Try the definition there. Breaking down of glucose in the absence of oxygen. Remember, anaerobic respiration only occur in the cytosol. So the enzymes for anaerobic respiration are also located at the cytosol. Okay. Right. Now I am going to talk about the steps of the anaerobic respiration. Are you all done? There are 2 types of anaerobic respiration. Only one type is happening at one cell type. Okay, not both happening one cell type. Okay. For the anaerobic respiration also, glycolysis is a common step. Therefore, glucose 6-carbon is broken down into 3-carbon pyruvate 2 molecules. What will happen at the beginning? 2 ATPs are utilized to activate the substrate. And what will happen to NAD plus? Will be reduced into NADH. And also 4 ATPs are produced. Am I correct? So this is glycolysis common to both aerobic and anaerobic respiration. So this does not depend on oxygen. Even in the presence or absence of oxygen, this is happening. So in the presence of oxygen, what will happen to the pyruvate? That is being transported to mitochondrial matrix. Now here, since there is no oxygen, this is not going to get transported into the mitochondrial matrix. In one pathway, pyruvate is converted into lactic acid, which is a 3-carbon compound again. So they are using NADH produced here. So here it is oxidized by removing hydrogen. While this is oxidizing, what has happened to pyruvate? Pyruvate is reduced to lactic acid. This is oxidized, H pluses and electrons are released. So pyruvate is oxidized to lactic acid simultaneously. Can you understand? So this reaction we call it as lactic acid fermentation.
And another types of cells, the pyruvate is converted first into acetaldehyde, which is a 2-carbon compound. Therefore, carbon dioxide has to be removed from the substrate. So then acetaldehyde being converted into ethanol. Ethanol is again a 2-carbon compound. That's the final product. So here they are using NADH produced at the glycolysis. So acetaldehyde is reduced to ethanol here. You can see that NAD plus is again produced. They can be used again in the reaction. So kind of recycling of NAD plus is happening here, which was not happening in the aerobic respiration, right? So what is the name for this pathway? This we call it as ethyl alcohol fermentation. Not ethanol fermentation, ethyl alcohol fermentation. Understood? Ethyl alcohol fermentation. This is usually happening in fungi like yeast and some bacteria. Lactic acid fermentation is happening in human muscle cells. For example, after exercises, vigorous exercises, we get cramps. The cramps are due to lactic acid produced in the cells. Also, lactic acid fermenting bacteria. Both these processes are used in industries nowadays. So what are these industries? This one? For which industry we are using this reaction? Milk products. What product exactly? Yeah, yogurt, cheese, using this method. So lactic acid is coagulating milk so that you can produce milk, the yogurt, cheese, curd, those things, right? And here, for what industry we are using this one? Alcoholic beverages such as wine, beer, toddy, and all that. The final product is used. Also in bakery industry, the carbon dioxide release here used for rising the dough, right? Okay, understood? Right. So what are the products here in the lactic acid fermentation pathway? Lactic acid and ATP, no? This is reused, no? And then here in this one, ethanol and carbon dioxide and ATP. Only 2 ATPs are produced in this pathway compared to the 30 ATP or 32 ATP produced in aerobic respiration. So this is usually done in organisms which are very simple, less energy consuming, right? So you can't use like in the complex organisms these two pathways, right? Then other, right? Hari. First of all, read the book completely without writing anything. See if you can understand. Yeah. No. You have to write cytosol here. Will you read? Now write this one, everyone. Right. Try the topic now. Lactic acid fermentation. Lactic acid fermentation. Lactic acid fermentation. Write the given description there quickly. Those are definite questions coming for examination. So you need to remember every point as it is in to write in the exam. Okay. So these things you can't change and write. Okay. Write down this. till lack of fermentation also you can write.
All of you done? Write down these two questions. First question, write the differences between aerobic respiration and anaerobic respiration. Always try to write best answers first, okay? Number 2, write the differences between lactic acid fermentation no, just 4-5 lines. And ethyl alcohol fermentation. Quickly write the answer. See if all the pages are printed, Pute. I have not put the numbers, just go through and see. Start writing the answers now. Yes, Upeki. Hmm. No, two different. Yeah. Acetyl group is maybe related with this one, but it is combining with coenzyme A to produce acetyl coenzyme A. But acetyl group must be related with acetaldehyde because two carbon both. You're finished? No? Continue. Right. What is the best thing to write here? Hmm. Yeah, occurs in the presence of molecule oxygen, right, as it is, okay? What is the second best one? Yes. Comparatively, large amount of ATP is generated. Thirty O, 32 ATP. This side, comparatively smaller of ATP is generated. How many? 2. Right. What's the third best one? Just another one best thing. Final hydrogen acceptor or electron acceptor is molecule oxygen here. This case, if it is lactic acid fermentation, what is the final hydrogen acceptor? Pyruvate. And if it is ethyl alcohol fermentation, acetaldehyde. Yeah. Molecular oxygen
You can say that it is an inorganic compound also. In the comparison, you can take as separate point also. So these both are organic compound. You can say what you said now. Number 4 occurs in both cytosol and mitochondria. Here only in the cytosol. Anything else to write? That's all. Final products also you can compare here, water and carbon dioxide. Here, if it is lactic acid fermentation, lactic acid and anything else, and ATP, no? Here, that one also ATP is there. Huh? No, that is reused, no? Right? Here, final products, ethyl alcohol fermentation, ethanol, carbon dioxide, and ATP. Right, write that too. NAD is reused. Yeah. Right. Now everyone go to the question tube that I have given to you. The separate question tube that I have given. We quickly do one question from no that yeah, that one. If yeah, the first page is having a question from aerobic respiration. So we'll see whether you can get the answers. Do the first page quickly. I'm giving only few minutes. Okay. Everyone having the tube? I'm talking about separate pages that I have given online students. Also, I have posted the tube to you in the group. Not that page, Pute. Turn, I'll tell. Not the first page, neither. It's not first page. Turn. The last page, ne? The last page of it. Let me show. Yeah, here this one. The question from 1 to 4 are based on the following sketch showing the cellular respiration. This one. Quickly do. And at home, you need to finish all the MCQs and remaining questions in the photosynthesis and respiration tute. Okay. All right. Shall we go to the next question? All done? I didn't discuss the second part of this question. Eh? We'll write quickly the answer for that one and go to the given question now. So, lactic acid fermentation and ethyl alcohol fermentation. What are the differences? What are the differences? Carbon dioxide is released. So here carbon dioxide is not released. CO2 is produced or produced, also you can use. And then final product is 3-carbon lactic acid. Final product is 2-carbon ethanol. Final electron acceptor, hydrogen acceptor. Pyruvate is here. Here it is acetaldehyde. I didn't write the full sentence. Is that all? Here, 2 steps. Right. You can write example of organisms also in the both side as it is given in the book. And did you do that question given? Let's go to that one. Name the processes indicated P, Q, R. What is P? Glycolysis. Q? Krebs cycle. No, not link reaction. The Q is Krebs cycle. Cycle is there. R? Electron transport chain. State the sites where PQR occur in living eukaryotic cell. P, cytosol. Q, matrix of mitochondria. R, cristae of mitochondria. State number of ATP molecules produced in the process are one glucose molecule, 28, which write a complete equation to show the reaction which converts compound A to compound B. A to B, complete equation, the pyruvate coenzyme A plus NAD plus, converting it into NADH, acetyl coenzyme A and carbon dioxide. Yes, balance it also. And here, name X and Y shown in the above sketch. What is X? Did you find it out? NADH, because it's producing Krebs cycle and glycolysis and pyruvate oxidation, all three. Then Y?
FADH2. Name the two major end products produced utilizing the compound A in different metabolic pathways in the absence of oxygen. Lactic acid and ethanol, right? Yes, good. Then come to this question also. There's a big diagram in that X and Z and so those things. Let's see whether you can do that. Quickly do. Y cycle is there, Z cycle is there, X is there. Do quickly. Shall we discuss? Hmm. Few metabolic processes are given in the above sketch. Identify processes 1 and 2. What is 1? Zero respiration. What respiration? Aerobic respiration. You have to add that part. That is a mandatory part. Right? Wrong. No. We don't know whether it's anaerobic or aerobic. So this is definitely aerobic. No? Write the specific answer. Right? Okay. And next one. 2. Photosynthesis. Calvin cycle. No? Photosynthesis is fine. You can take. That's okay. T3 mechanism is also fine. Calvin cycle. Yeah. But photosynthesis should be there. Next one. Name the steps X, Y, Z. X is glycolysis. Location is cytosol. Y is Krebs cycle. Location is matrix of mitochondria. Z is Calvin cycle. Location is stroma of chloroplast. Right, nice. Next one, name the compounds indicated in 4 carbon in the process 1 and 3. So 1 and 3 carbon in process 2. Process 1, 4 carbon. Oxaloacetate. 3 carbon in process 2. There are 2 3 carbon compounds given there. Yeah, 3PGA as well as G3P is also there. Right? Process 1, 4 carbon compound is oxaloacetate. No. 4 carbon in process 1, 3 carbon in process 2. Read the question one more time. Okay. Did you write the answer, everyone? Uh, 3PJ and J3P. Right? Next one. State the enzyme which converts the compound 5 carbon into 3 carbon, Rubisco. State 2 characteristics of an enzyme which can be explained by the mechanisms of the enzyme activity. Yeah, enzymes are highly substrate specific. Enzyme catalyzed reactions are reversible. Any of the suitable answers you can write it here. From the steps X, Y, Z, which steps produce ATP? X, Y. Yeah, that's all. Alright, now quickly come to the last chart in the textbook at the end. Respiratory quotient I will be doing along with the experiment little later. Now I'm going to the chart given there in the textbook. How other substrate enter into the respiration, cellular aerobic respiration. So we usually tell about glucose, no, isn't it? Other than glucose, so many other substrates can enter into the aerobic respiration pathway. So go to the chart. That is in page number 71. So carbohydrates other than glucose, you can say that starch and glycogen-like substrates converted into its monosaccharides and enter into the glycolysis. You have to tell the step which they enter and what is the substrate. And then you can say sugars such as maltose, lactose also converting into their monosaccharides and enter into the glycolysis pathway. And other than the sugars, and there's one more arrow from sugars to glyceraldehyde 3-phosphate. Make it one arrow, sugars to glyceraldehyde 3-phosphate. It shows some unclear diagram there. It has no relationship with amino acid. Sugars to glyceraldehyde 3-phosphate directly.
So glyceraldehyde 3-phosphate also is an intermediate in the glycolysis pathway. You can say some sugars may enter as glyceraldehyde 3-phosphate into the glycolysis. If it is a 3-carbon sugar, they can go like this. And then other than carbohydrates, proteins and fats are also used as substrates. Then fats are converted into fatty acids and glycerol. Glycerol can enter into glycolysis as G3P. Fatty acids can enter into Krebs cycle as acetyl coenzyme A. And then proteins are converted to amino acids. The first thing they need to do is to remove the amine group from the amino acid. You are removing. And then it can enter into glycolysis as pyruvate and into Krebs cycle as acetyl coenzyme A and directly into Krebs cycle. Can you understand? Hari? Go home and draw this one more time and try to write this in words. Okay? This is usually like some are ignoring this one, but I have seen so many questions coming from this section. You should not ignore this section. Okay? Next week, there will be the examination from organelles, meiosis, mitosis, and goals and cancers, those things, right? And we have 1 hour remaining. I'll be coming to the class and sometimes I might do the lesson or else I'll be doing some practice questions for the next coming up exam from photosynthesis and respiration. Okay, I'll let you know about that 1 hour. But 2-hour test will be there. So everyone be ready nicely like in the previous one that you performed well. This one also, write some essays, read the short questions that we did and come with the proper practice. Okay, so everyone, good afternoon and have a nice day. We'll see you on next week. And do the questions and come in the tute. I have to discuss the MCQ also, neither. Will you bring that MCQ on that day? Everyone, don't forget to bring the MCQ.